Hi, Jamie. How are you?
Hi, Katerina. I'm well, thank you. How are you?
Good, good. Thank you. We'll wait a few minutes until we start.
There we go. Having a good weekend?
Hi, Victoria. How are you?
Hey, good morning, Katarina. Good morning, Jamie.
Hi,
And
Victoria. How are you?
happy. Glad to be here. How are you
Oh,
both?
I'm well, thank you.
Good, good. Thank you. We had a lot of guest speakers last week, so a lot
Every
to discuss.
every time we have a weekly recap, I always go, "Wow, how much ground have we covered in a week? Wow."
Yep. Next week will be less busy, gladly. We'll have one room a day, and let's see. I always try to, since it's kind of scheduled ahead of time for a while, to get confirmations again from the speakers. If I don't get them like on the day before, I'll cancel the room. But let's see, maybe I'm too I'm too worried always that people won't show up. I mean, the room is just about, the person and their research. So if they don't show up, it's kind of, you know.
Yeah, the momentum you've picked up here is phenomenal, Katerina.
Thank you. Okay, let me pin the first link, and here we go. So yeah, this was an exciting week. As I said, we covered a lot of topics and learned a lot of new things from editing gene expression very precisely to quantum physics, like nuclear physics. So BCI, novel BCI technology, it was really amazing. And the first speaker we had on Monday was like a wonderful start. She was an amazing she is an amazing public speaker. So let's talk about her first. It's Dr. Shiri Levy. She's at the University of Washington, and she is a researcher there. And she was also back in until 2000 a surgeon in combat engineering in Israel. So yeah, she has kind of a diverse background in life and in from the work perspective. So she used to design and implement combat ready training courses for soldiers for intensive exercise, and was awarded for the best soldier and best instructor by the president of Israel. So yeah, she was a really great, amazing person in the Israel Defense Forces, and now she's a researcher. Yeah, her talk was about. You can see the post. It should be open access. So she developed, or she and her colleagues developed, process in very precisely changing gene expression of precise genetic code, and she did that with 4 genetic regions, and also additionally, while doing this, she also revealed inhibitory function of the Tata box that is actually in a quite distal promoter region of these gene codes that she tried to implement. So what they generated a computer designed PRC2 inhibitor that fusions with DCAS9, and by doing that, they could, as I said, very precisely change gene expression of precise locus using gRNA. And they did that with these 4 different genes: TBX18, P16, CDX2, and GATA3. And this resulted in the precise H3K27Me3 and EZH2 reduction of gene activation and functional outcomes of in the cell cycle and the differentiation of the cells. So so they identified this, or they computer generated and designed this protein, this EED binder, and they named it EB, that basically can very precisely, in combination with DCAS9, manipulate gene expression levels of precise loci. And the cool thing about it is that with this, you don't change the genetic code itself. And additionally, you can design it in a way that you need to take a drug, a starter drug basically, in order to to activate this designed protein and this whole cascade of genetic function, of gene expression function. And when you stop taking that medication, this activation of this gene expression function will also stop. So you not only have that way a very precise way to control specific gene loci, so very specific genes, but also you can become quite precise in the time, because you can link it to an activated drug like this. So this is really great progress for precise manipulation, let's say, for novel cancer drug treatments and other kind of gene therapies that don't change the genetic code. And another interesting or insight that she gained through her research was that you have this inheritance of epigenetic mechanisms that she showed in this stem cell when they proliferate up. So she saw this activation of these genes during very specific days, on day 3, for example, and that this function was inherited So it's a also additionally quite interesting basic science insight that she gained during this designing of this method, and then yeah. And I think she explained it really well how epigenetic works. So epigenetics, just to quote her, if you imagine all the different cells you have, liver cells, skin cells, and so on, they have all the same genetic codes in them, but they look very different and they have very different functions. And this is thanks to the epigenome. So how the genes are being controlled um, to turn them into different cell types and with different functions, doing different jobs basically in the body. So I don't know if you have anything to add. I think this is just to give you an overview of what the research was about. If you want to have a deep dive, please listen to the replay or go to the paper. Yeah, go ahead, Serena.
Yeah, I thought there were two really cool things about it. The de novo designed protein, that was for one, because that was completely computationally designed, and I always like hearing really good applications for these design proteins. But I remember at one point the room got completely locked on to the 3D implications of this distal site, 500 base pairs, and what that might say for the the geometry of the chromatin in that case. She didn't have answers. I guess that's not was anything that she was looking at. But it was interesting to see just how the room kind of locked on. But it'd be interesting to hear more about 3D structure of chromatin and when we're manipulating expression like this. It's kind of fascinating. But I actually missed the part that what was it, the foxo something, the drug that it was activating. I missed that it was,
FOXP1.
yeah, the
Yeah.
fox. I missed that there. Um, so I'm glad you went over that, Katerina, because I didn't know they also had that additional level of control. Did that activate the designed protein, or was that activating CASP, or what was it? Do you know that its binding mode?
Yeah, so when they put this construct basically into the cells, you are very it's became it became quite common now that you basically link this. You kind of have to express this design protein, right? And in order to do that, you can link it to being activated by antibiotic, for example. And that's what they did here. It's becoming relatively common to do that, to have this additional control. Yeah, but like in animals, it's relatively common now. So you see a lot of papers where that's the case. I think David Sinclair and others also use that. So you have like this regulation that you can stop basically the activity, because at some point, especially during development, which is kind of here, right, when you turn stem cells into something like she did here, you can this is kind of a time precise mechanism that has to stop then at some point. So yeah, by using that, you kind of, when you stop taking the drug, the gene expression of that design protein stops, and then the function stops.
Didn't this have the kind of potential to take, like if someone was having cancer in a certain part of the body, and this is the potential to actually stop those cells from replicating or something? Am I misremembering? Like the potential of this was huge of what
Yeah,
it could do.
it's huge. And but the Fox G1 is the next paper. I'm not sure if
Hmm.
you're the right one. The pure proliferation in the progenitor cells of the human forebrain, that was the next speaker. But still, she also looked at more at differentiation mechanisms than proliferation. But yes, you can basically with this target then cancer cells, but also other gene loci and stop this or activate this different kinds of gene expression mechanisms. So yeah, I think you're thinking about the next paper about the proliferation, because that was really we can go there now if you want. But the Tata box, what Serena said, I wanted to mention that was really interesting. It was shortly just mentioned the G4 formation in this paper, but yeah, she didn't really go into that. I guess it's not that trivial to look at these formations in the living cells yet. So you have for these 3D formation, for specific ones, in cancer, there have been developed drugs that target these specific formations, and then you have antibodies that target very specific formation, mostly G4, and now also the eye motif. But not all formations, and not in like a living. It's still hard to do it in a living tissue without fixing it, and sometimes fixing it just disrupts these kind of formations. So yeah, but it would be really interesting to look into that. I agree.
Oh, you mentioned the eye motif. Could you briefly tell us what that is? And you had a hypothesis paper about that. What was the right?
Oh, sorry.
Didn't you?
Yeah. I had so basically what this is, you have eye motif and G4, so you have specific specific gene sequences that basically form this T or cross structures, and they have been shown in different like not in vivo. So what they basically do to find out if these formations are possible is to um, change pH, change different types of factors to the DNA and see what the DNA does, like how it changes. But usually it's not something that people have shown in the living cell. However, the G4 formation and also the eye motif formation had been shown then in vivo, in like these cancer cells that you keep in the lab. And the interesting thing about that was that different ion concentrations such as magnesium and K plus change or affect this eye motif formations, and also indirectly through these eye motif formations, G4 formations can also be manipulated. The interesting thing about that is that these ion concentration fluctuate quite a lot during synaptic plasticity, long-term memory formation, longer excitability changes, even in shorter ones. You have this flux of ion concentrations, and NMDA receptors need also a higher magnesium to unblock. So, and those are highly involved in long-term memory formation. So that's why I wrote this article that maybe especially this eye motif formations play a role in synaptic plasticity for gene expression changes. So yeah, I wrote a hypothesis paper about it. I applied for grants for this. It was a few years ago, and I never really got the grant. So I just wrote this, and then I thought somebody else can do it.
And they did. Huh?
Yeah, that was interesting, especially the G4, and now also there's some emotive work in vivo now. So yeah, so that's interesting. I got a lot of invitations because of this hypothesis paper to speak in conferences. So that's what came out of it, basically.
It's quite exciting when you see a paper that you wrote something about actually saw some activity later on, eh? That it's, still paying off down the line, even if it's not in ways you expected.
Oh yeah, I don't. If somebody else, I'm just glad. I'm just curious to see if it's true or not. Who then in the end does it? Doesn't really matter in the end, I guess. So I'm just glad that people work on it because, as I said, I think before the cancer drugs are way more evolved. They always had more money. They are very, very specific cancer drugs that target these functions because this was always hypothesized that these are regulatory mechanisms in cancer. So they are pretty quick in developing these very specific drugs that target these. And I always think as long as it's a small molecule, we could also use them in severe mental health or, brain development and so on, disordered. And probably not even for, you wouldn't need to take it chronically. So, yeah. So that's my hope. If I would, get the money I applied for, that was my idea a bunch of years ago, to just screen with, computational tools through all the cancer drugs and screen to all the gene expression data that's available at NIH and pair them with novel cancer drugs and then try those treatments out. But let's see, hopefully somebody will do that at some point.
To everybody listening here, this is an example of a pure scientific mind here with Katerina. The desire to have growth and betterment for everybody and for ideas to come together to make everything better for everyone. So please remember that and subscribe and listen to all of these replays because they're all worth listening to. That's my take.
Well, it is quite commendable that in the face of such competitive funding, when a project doesn't get funded, that you at least had the energy and resolve to put it into a hypothesis paper so others could pick up. That is quite commendable. So great.
Oh, yeah, thank you. It wasn't, I don't know. I do silly things out of frustration.
I mean, whenever I
Um,
got grants denied, I was just pissed and just moved on to something else. I can't imagine having enough motivation to actually put it into a hypothesis paper. That was good. Hats off.
those, but
Definitely remarkable.
ResearchGate, because they kind of got annoyed with me. There are not too many hypothesis paper places, and you can publish for free, but they don't. But they want you to actually pay for the open access. But I said, yeah, I didn't even get a grant for this. I don't care. Like, I want to publish it so people. I'm not paying a few $1,000s to publish this without the grant. So I just ended up putting them into bioRxiv, whatever, and then put it on ResearchGate. So I had a few, but for those I didn't put as much. Like I wrote it together. It's a lot. It's not condensed enough, I guess, for people to. Well, it has a bunch of reads, but like the immune system, neurosystem, coevolution, and why that would help us to find new drugs and treatments. It's so long, that paper. It has all this stuff. Like it has the everything that's immune system and neural system discussed there, and how to screen for it, and all this stuff. And the hypothesis is that you would ideally in the future do RNA sequencing on people, and you would recognize, like ideally with buccal swab, like but this was years ago where I did that. And then you would basically recognize, okay, in that person at some point you should stop the communication between the immune system and the neural system to basically prevent things like Parkinson and dementia and ALS and so on. Because from my idea, it's just that those two systems keep inflammating each other and don't stop. And if you at some point would just take a pause, you could probably at different time points in life prevent maybe those disorders. But anyways, it's off topic. I'm sorry. Let's let's go to the next one. Next we had Carl Ernst. He was here and gave also a really interesting presentation. He's an associate professor in Canada at the psychiatric genetics at the McGill University. And he talked about this FOXG1 and the dosage basically of FOXG1 expression tunes the cell proliferation dynamics in the human forebrain progenitor cells. So basically, if you have too much of the FOXG1, it's not good. And if you have too little, then you basically get cancer, and quite aggressive brain cancer. If you have too little, you get quite significant developmental problems. So I think it was really interesting how he approaches the studies. He works really closely with his patients, and they basically get cells from the patients and grow them, like stem cells from the patients, grow them, and look at the iPSCs derived forebrain neural progenitor cells of the patients, and basically check what is going wrong. So they as I said, they had the cells from the patients, and then they saw that basically mostly pathogenic mutation in the FOXG1 affected the cell proliferation in the human brain, and that basically led to the developmental issues in the patients. And with that, you can basically, in the future, hopefully with that approach, you can hopefully in the future prevent disabilities. Like, this is a rare disease research, but yeah, this shows how also rare disease research can turn into a really broad, applicable, beneficial treatment at some point for probably different disorders too. So yeah, I don't know if anyone wants to add to this.
he was a remarkable man when he was up talking, because not only was he doing this incredible work, but he was actually driven by a great deal of compassion, as he'd been working with like handicapped children earlier in life, and he was even making songs for children and things like that. He was driven by a quite a fantastic drive, and didn't he say that the Fox G1 was kind of like interesting because it seemed to be so powerful that the body seemed to almost be keyed up to shut it down as quickly as possible, because its effects on the brain were that powerful that even exposure for a short period of time was causing certain dramatic changes. So it's like a some kind of super thing. What was it? Yeah.
Yeah. So yeah, if you express too much, if it's been activated too much, it can turn into a very aggressive brain cancer. Yeah. And if you have too little, you basically don't have enough brain cells, and then you have basically too small of a brain. So yeah, it is quite powerful, and it has a very important regulatory mechanism, especially in the brain. Yep.
And he had said that the research he's done at the moment still leaves a lot to find out in the future, right? Because finding the right cases and being able to do the right kind of tests is difficult and long and time consuming. So there's still a lot to uncover there. But the fact that he was even uncovering what he did was some quite big steps for potentially other people in the future too.
Yeah, exactly. Yeah. So the exact function now, he's working on that, like how it changes proliferation, and exactly, and what exactly in the cells happen. But yeah, this was one really big step, and also he wants to basically have tissue available in the future that's easier to collect. So it's very similar to the cancer, the problematic of collecting tissue and making it cheaper, basically the whole process. Like in the cancer research we talked about later in the week. Yeah.
I like hearing stories of pursuits of rare diseases that uncover much more general things. I at least it used to be. I'm not so sure whether that's still a factor, but I imagine it is. That the pharmaceutical companies would be less interested in rare diseases because of the bottom line and the market size and things. But I, 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 I always like hearing, because often when an investigator goes after a rare disease, well, I don't know how often, but that it's not uncommon that there's a personal motivation and there's some definite passion behind the work. And when we uncover things that are more broadly applicable, it's a real win.
Yeah. And the funny thing is, he also never wanted to actually go to science. Like, um, like it was not his main career goal, and then he just wanted to solve, get different answers when he was at the university, and then the professor told him, okay, then you have to go to neuroscience, neurodevelopmental science, and that's what he did, and it was interesting.
Definitely, to whoever is still tempted to go and listen to the replays, at the absolute least, hearing the backstories of these people is remarkable enough to give a few minutes of your day towards listening to. and the rest will just follow. Your curiosity will just take you there. The how they got there is always fascinating, always.
And then we had Dr. Gilbert Gallardo here. He's assistant professor of neurology at the Department of Neurology in Washington University School of Medicine. And he works a lot with Alzheimer's disease, neurodegenerative diseases that are in alpha-synuclein and tau related. And yeah, I pinned the paper here to the room where he basically talked about I could also put the present. If you want to have the presentation again, please contact me. I can send you the link. It was a really pretty presentation about neuroglia. You would have loved it, Serena. I think you were not there, were you? I'm not
I
sure.
know I'm missing all these good ones. It's terrible.
He started his. Let me go and get
Oh wait,
the.
I think at least one of them he had an astrocyte and microglia story, and then there was a network about them that was less appreciated. Normally, it was thought, is this that talk?
Yeah, let me. He had a very pretty presentation about
Mm-hmm.
the glia history. So there you go. The glue that holds the proper nervous part. He went a little bit into history there when the first research was done on glia, and then he went into a little bit of the research he had done before about the MST FOXO signaling pathway, how it mediates oxidative stress, and if it's phosphorylated in these SODTG mice that have this motor neuron degeneration. So he went into that. So his presentation was really good because he gave us a little bit of insight on the path to this paper, basically. Then he talked about alpha adductin, ATPase complex, how it's associated also not just with these accumulations, but also with hypertension, and that it's predominantly expressed in primary astrocytes, especially in the spinal cord. And so he proposed back then a mechanism for motor neurodegeneration in ALS. And then he started his research in astrocytes and microglia, how they impact motor neuron degeneration. And further on, then he showed work of knockout mice with this alpha NKA mouse model work he did before, and then he came into the Alzheimer's disease. So he gave a really good overview of his research a little bit in general, and how alpha 2 NK is upregulated in Alzheimer's and tautopathy mouse model, and how he did a pharmacological inhibition of this mechanism, and he could prevent these tau regulated pathologies. And it's actually a drug that is being used for hypertension or cardiovascular related disease. So this was basically, he said, what most people are interested in, like the science news world, because he used a drug that's already FDA approved to basically prevent and also downregulate the the severity of Alzheimer's disease with this drug. Yeah, I think it was a really great talk, because he really made an effort to give us like a broader overview and of the different mechanisms he looked into. So it was really good. Anything, does anyone want to add to this? If not, we'll move on to Bruce Hansen, Dr. Bruce Hansen. He is a professor of psychology and neuroscience at the University of at the Colgate University. I'm sorry. And yeah, his work, let me see, I have to put the link up. Sorry, I have to find the right link. It was a lot of stuff. So his work was really interesting, more in network systems neuroscience field and in humans. There's the right paper. So he is interested in everyday visual experiences, and how the operating, or how these senses, like especially the visual system senses, are computed and processed in humans. And he used this imaging system, DETI, dynamic electrode to image mapping. His presentation was also really, really good. I don't know where I have the link. Oh, should I put the presentation up or the paper? What do you guys think?
We could do the presentation too.
Yeah.
I remember it was really, this was the visual field mapping, right?
Yeah, exactly. Let me put Bruce Hansen. Can't find it. Oh. Why can't I find it?
Because of how much I've covered this week.
I must have somehow deleted it by accident. Let me.
Well, we have the paper up.
Yeah, we have the paper up. Anyways, yeah. So he really explained very well how they performed the data collection. They basically had the stimuli, normalized images, samples from database of scenes, and they then collected from a lot of electrodes the neural data, whereby the participants viewed the stimuli, and they were recorded by, like, they had 128 EEG channel recording, and by different filtering processes, and they did time resolved principal component analysis of these filters. They basically were able to do a quite specific image analysis that could map these specific channel inputs to this image specific analysis and reconstruct basically the image. And what I think was really interesting about this work was how they looked for the time constant also. So basically they saw that the attention, or basically what is being tracked in an image, varies over time, and it changes. And I think that is really important to know, especially also hopefully once he goes back and looks into different mental health, patients with different mental health disorders. I would assume, and we discussed this, it's just a lot of work, and getting more patient and more data and going through the data is really a lot of work. That I would assume that this could be a really good way to check for different mental health disorders, or maybe an onset of different neurodegenerative disorders, that this neural response over time and tracking an image is probably different in people with different disorders. But there's still a lot of work to be done, basically. And just the fact that he found this and tracked this over time is really tour de force, how Serena said his work. So it's really interesting.
Well, it was because it was so interesting that there was such a marked time dependence on even on a per electrode basis, and it was the time frame was over a quarter of a second. But when they showed images of where even just the first principal component, he said that explained most of the variance, was focused on. Sometimes it was fine detail, and other times it was the broader features, and that and it was never, it wasn't clear whether that reflected the any kind of attentional ordering in the experience, or whether that was just being buffered, sort of buffered up in the visual field that would be selectively filtered with attention later. But it was really fascinating how much time dependence there was in that data. It was it looked like there's just a lot of stuff there to mine.
Yep. If anyone has more, if not, we can move on to the next one. I really did something to my notes. I kind of jumbled them up. I'm sorry that this is going. So the next room was our nuclear physics room. Let me put the link up. This was quite.
This was crazy. Yeah, I just blew my mind.
You want to go ahead and share?
Well, so it was. I had no idea this was going on, and as it was a high energy particle physics experiment or series, and from what I gathered, and I was just really blown about how, as the energy of these protons are speeding up, the gluons are just spraying everywhere and becoming in this dense matrix of quarks and gluons, and how a treatment that doesn't involve entanglement failed to explain them. And really the emerging result was that to get a handle on the entropy of this system, and I think he was, the author was so humble in what he was presenting. He said the interior of protons, but at the end he followed up with, well, yes, and of course the rest of the universe works this way too. Yeah, this proton is that simplest form of matter. But in terms of, and at cold temperatures, room temperatures, I didn't get a chance to ask him how much of this is going on at room temperature chemistry or body temperature. But just the fact that there's all this stuff going on in a single particle, which we think of as a single particle, which isn't. It's three quarks and a bunch of gluons, especially as you accelerate them to higher energies. So actually capturing a formal expression for the entropy that is consistent with it being maximally entangled, it was mind-blowing, because I just, as a chemist, I normally think of, well, that's as far down as we dare to look, and okay, nuclear chemistry, but we're not splitting atoms here, so it's all quiet down there. No, 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 no. All kinds of things going on, particularly at high energies. So it was just a fascinating. I was mostly speechless, but I wanted to ask him. But I feared the answer was just going to be that it's not as important at the low energy, that we live in. But it's still there. It's still how nature works, and it was an amazing commentary on the universe, I thought.
So, Serena, just to ask you, so when you cool down, I don't know, I know that people always say this might not be going on at our, at room temperature or, regular temperature and so on. But isn't it, I always ask myself, isn't it cooling down just slowing down the process so we can observe it? At higher temperatures, it's not observable for us, maybe. Or is it really the case that it's not then going on? I don't see how. Like,
So that's that wonderful quantum world way down below. It's not just slowing down. It's that there's the discrete energy levels that are no longer populated without sufficient energy, or it just becomes lower and lower probability of ever having any population in the excited states. And so, when you get down to those low energy states, that they just may not be populated. If you excite them in any sense, they're still there. They're available, but they're not populated, or just populated much less probably. So in that case, when he showed one of the diagrams of in a cold system or a low energy system, and there's like one or two gluons flying around, and then the high energy one, it's this whole gaseous dense matrix of all kinds of crap flying everywhere. But those are quantized states, and they're populated with a distribution depending on your energy. And so in those really cold states, and that's what I really wish he sort of had to go all of a sudden, and so I didn't get my question in. But I was wondering, how much, at our room temperatures, how much of this is going on? Because I don't think what was so interesting to me is I don't think this is appreciated outside of nuclear physics at all. I mean, what do you mean entropy inside a proton? What are you talking about? And so, I was thinking, what, between the proton and the electron? It's like, no, he's talking about in the nucleus with the quarks and gluon, high energy stuff. So it was just really, it just blew my mind that there's all this stuff going on down there that may have little relevance to everyday chemistry, but it's just how nature works in higher energy situations. So.
yeah, so what I loved about this was, I can just read a quote that our study shows that the interior of a proton seen by a passing photon must be entangled with the unseen part in just this maximal manner. In practice, this means that we have no chance of predicting whether, due to interaction with the proton will decay into 3, 4, or any other numbers of particles. And what this means is that basically these experiment results, the universe is non-deterministic. And I asked him specifically again the
I
question, does
remember
that
that.
mean the universe is non-deterministic? He said yes. So
Well, I
there we
remember
go.
you
We
latched
proved
onto
that.
that. There was another quote from a physicist that I remember that quantum mechanics will predict that the probabilities are deterministic, but not the outcome, which I thought was an interesting way of speaking to whether the universe or whether what did you call that? You had a great phrase, dork puppets. Whether we're all dork puppets.
Yeah, exactly. Are we all puppets on a dark puppets on a string or not? And apparently we are not.
And so quantum mechanics will predict probabilities are deterministic, but not actual outcomes.
Very exciting work, yeah.
Okay, so later we had on the same day a room about cancer research. And I know the title was a little bit, well, it's actually a title that he agreed on. So he started the presentation with that most people that smoke, almost 80%, this was Zhengqing Huang from Albert Einstein College here in New York City. And most people that smoke actually don't get cancer. And he showed that smoking, of course, increases the rate of mutation. And what they did here was, and this is quite expensive to do and quite hard to do, they did single cell analysis of somatic mutations in bronchial epithelial cells. So they collect from biopsies this tissue, and then they had also control group from people that don't have lung cancer. And then they look at single cell mutations in these lung cells, and they found that people that smoke have more mutations. At some point, actually, with the time and amount of smoking, there's kind of a cap. At some point, you don't get more mutations if you smoke more. But still, there is a relatively high percentage of people, although you get this higher mutation rates, that don't get cancer. And this is basically just a beginning of this sort of study. What the goal is of this research, and I think it's a really interesting one that I really like, is to see what are basically, he didn't name it resilience, but what are basically the resilient mechanisms of these people that smoke but don't get cancer, and can we study that and leverage that. And also at some point he wants to also look at what smoking does to the germline and other cell types in the body. So this is basically a really great start to explain the methods. They also did some technology development, and he will come back with his colleague maybe in the summer to discuss the technology they developed to do this quite efficiently. as because I think the technology itself is really interesting, and yeah, so this is basically an ongoing research to figure out, and I think it's a great model because uh, it's a really significant impact to smoke, right? For years. And you could go into studying people that live in cities versus people that don't live in cities, but not all the cities have the same rate of pollution. Not even living in the same city, you're exposed to the same level of pollution. So I think using smoking versus non-smoking as a model here is really it's a really good one to detect this protective cancer mechanisms in people that people have and then other people don't. So I think, yeah, I think this will hopefully give us some data and maybe treatment options or even preventive, like Jamie asked if he can basically develop a vaccine for cancer at some point based on the research, and that would be really exciting, but it's still a little bit far off. And also they are looking into different tissues that they don't have to rely on biopsies because it makes it more difficult to have higher sample rates. So they are starting to look into blood and buccal swab tissue and see if they can basically replicate this, so see the mutations also in these tissue samples. And if they can, that will, enable them to have higher rates and then look at subpopulations and, have a more granular data analysis. Because right now they don't have the statistical power to look into different lung cancer types, different ages, male, female. They just don't have the statistical power yet to do so. But with going into those tissue samples, they could in the future.
That's right. I remember asking that question, and the fact that he did say that eventually that would be definitely an avenue that they would be going down. The idea that there even is a path like that baffles me, that he's not being drowned in funding money, considering the death rate of cancer patients and lung cancer. Just baffles me. But the idea that he's working on it, and even if it is early stages, an early stage is still a stage, and it's very exciting. So very much I'll watch this space kind of situation.
I was wondering, I couldn't tease out from the data whether in the talk that whether he was saying that, because he had, he showed graphs that were very clear for never smokers. The slope was still positive with age, and there was a very clear steep effect with smoking. But I was so preconditioned from the title. Why don't smokers get cancer? Was he saying, or was his data supporting the notion that the the smoking exaggerated an effect in people that were predisposed to those mutations, or is it actually causing these mutations in people that would not have, that weren't otherwise predisposed? I couldn't quite tell where that.
I don't think he knows the mechanism yet, why people, the full mechanism yet. So he just saw that people that smoke have more mutations also in cancer related, non-cancer related mutation signatures, basically, that correlate with smoking status, and were showed a stronger correlation. But actually the strongest correlation he showed was always chronological age. That still has the highest impact on this mutation signatures. So he compared smokers versus non-smokers, if you look at the figures, and what the mechanisms are that protect them from getting cancer. Uh, they haven't figured that out yet completely. So this was more like an analysis of the data, of the single cell analysis of these somatic mutations in relation to aging and smoking so far.
Yeah, I'm always curious if is it is it carcinogens from combustion products, or is it if you do a lot of grill cooking, are you also predisposed to those effects? Is it anything pinned to nicotine in particular? Does vaping materialize in these same effects? I'm always curious about that.
I don't think nicotine itself should be increasing
Yeah, I
cancer.
don't think it's labeled as a carcinogen.
Yeah.
Hmm.
It changes like memory and stuff like that. Acetylcholine pathway, it modulates it. But
But is there, I guess, I don't know if there's similar data on people that are around either campfires or grill cooking or, other constant inhaling of combustion products. I suppose the coal miners have issues.
yeah, coal miners have issues. So he chose this model basically, kind of a framework, or he's developing this framework with smokers. And for sure, people that work in coal mines or work, a lot of with combustion, maybe even uh, working with as dentists, you have a lot of those fumes from all the glues and all this stuff. Maybe even hair salon. They all have this, but I think he chose this model because just lung cancer is the most common cancer form, and smoking is, it's probably the more straightforward stressor that probably you can use. So, and that people get asked in questionnaires, right? If you were a smoker or non-smoker, there's no really medical questionnaire that asks you for if you grill a lot, if you work in a hair salon, and so on.
Sure.
Yeah, and then on Friday we had the talk, BCI talk, a brain computer interface talk. That was a group from Japan. They were really nice, and uh, they're neurosurgeons and also computational. So what they did was, they had patients with epilepsy that have these basically this implants in the brain. And so how it looks like, I don't know if you can see the image here. The patients have this electrode pads that record from a lot of cortical neurons, and in this case in the visual system. And what they did is, the patient was shown on a screen different images. Here on the paper figure was, for example, a bear out in nature, or a dog. And they basically then related that imagery with the ECOG acquisition. So from this electrode pad, the data acquisition, and recorded the data and reconstructed it basically into visual semantic spaces with high, and they saw that you could record high gamma powers, but also alpha and beta, and so basically they recorded that information, and what they did next was they gave basically the patients the task to visualize something else while the patients were still getting this visual cue from the screen. And this gave them basically a different representation of the feedback images. And they did that in a closed loop at some point, and basically the patients managed to increase the precision basically of that visualized, like inner eye visualized image signal to noise ratio. And the biggest contributor of this top-down control of the visual system was through the gamma band, high gamma band, which was, I think, really interesting to see, and what this will lead to, hopefully, into the future is patients that have kind of locked-in syndrome or ALS, and they cannot communicate anymore, that you can basically translate through the inner imagery that people are having into a signal that you can basically read out and communicate with people that have, locked-in syndrome or ALS. So they cannot, would not be able to communicate with you, but with this tool, they can visualize something in their mind, and you can basically display this on the screen for people. So I think it's really amazing that they could do that. They discussed that this is really, in this type of precision, only possible with implanted, so invasive, with this invasive method, to get like their inner mind imagery displayed. Yeah, so I think it's really interesting.
Wow, I missed this one, I think. But it's so they actually confirmed that if you are imagining something in your mind's eye, sort of, that does actually activate corresponding visual cortex fields, and that's readable.
Yeah, exactly. And then
Oh my God.
yeah, it was, and it's different from the imagery that you see on the screen. They could distinguish it, and with this feedback loop, patients could basically increase the signal to noise ratio to display a more, like a better and more precise image, basically through their neural activity, so they could control it. It was really cool.
Wow. So that kind of resolves a long-standing question, that when you imagine things, are you actually seeing them, or is that some other part of the brain that's holding the models of what you're imagining? So it's actually, we are repurposing the visual cortex for that.
Yeah, exactly. And there was a question, I think Victoria asked it, if we can basically, I think it was Victoria, what if people have this problem that they cannot really visualize something, can you still use that technology? And they said they just don't know. They didn't have a patient with that problem, so they don't know. They would have to test it, but yeah.
Well, and does that work with the same in the visually impaired?
Yeah, they they don't know. Well, I don't know. They are visually impaired that can still have imagery. But I don't know, Jamie, what do you think?
I'm thinking that to see any data on this, you probably have to see blind people who've never been able to see, because ultimately, if you've seen for a while, you still have the images in your head. If you don't have that disorder to not imagine things like that, but um, I think it would be fascinating. I personally, thinking about it, would be leaning towards that it actually still would show up. I'm just wondering, how was it you said that they map in? Like, how do you know if someone's thinking of, was it like, again, if you could think of an elephant, did they know it was like an elephant or an animal, or was it simply that um, think yes and it went to one part of the brain, think no and it went to a different part? I can't remember.
Yeah, they told them to have like a word, for example, imagine a word, imagine a landscape, different things.
Right, so they were calibrating that, right, in the same way that when people have done other tests in the past, they have to like calibrate it to what someone's thinking. So in that case, I would actually lean towards the visually impaired still could, as long as they've got a concept. But it would be really interesting if a blind person who was never could see, um, if that part of these parts of their brains would be activated. And I really appreciate you asking that question, Serena.
Well, it is. Yeah. I mean, if we are repurposing the visual cortex, those visual fields, um, and and what, that just that question: how much imagery have you been conditioned? Do you remember? Was that where the neural pathways were trained, or is there just something more fundamental that occurs in development? That if you're going to imagine something, you're going to reuse that architecture, and whether I can remember studies in cats about or kittens in terms of visual development, and ones raised in dark or delayed light, and there was I can't remember the solid conclusions, but the mapping was a little noisier, but it was the architecture was still there. Um, how much it functioned was a question of that. So the it remains an interesting question. Is that how the visually impaired would imagine things, or is it just a completely different remapping entirely?
It definitely would open it up to so many other questions, like if that was possible, could they then be, I don't know, taught other images? Like, could people that aren't blind or been blind since birth somehow interact with the world in a way that they could get these images put in the brain through other means or something? Just like all of these guests all the time. The exciting part isn't to find answers, it's to find ways to more questions.
All the good answers raise more questions.
Absolutely.
Yeah, so that was this week. It was a lot. Yeah, we learned a lot, discussed a lot. It was quite amazing. And the next week will be also really interesting again. We'll have tomorrow at 6 p.m. EST, Dr. Lisbona, um, how she could use sound waves to differentiate stem cells. She's from Australia, a researcher, so that will be really interesting. Then we'll have on Tuesday at 9 p.m. EST, Dr. Jim et al., like the group, will be coming and talking about quantum charging to shorten electric vehicle charging. It's a really interesting paper, the technology they use. So I'm really excited to hear about that. And then on Wednesday we'll have Dr. Timothy Brady again from Australia, and how he found that junk DNA could be actually the key to controlling fear. And then on Thursday we have another human vision researcher, Bechli Vanidis, and how human vision reconstructs time in different ways, and how it changes. So that's a really interesting paper too. And then we'll have on Friday at 10 a.m. Dr. Kwasniewski, and how peptides, building blocks of life, form on space dust. So yeah, that will.
That's going to be interesting. Yeah, it's going to be a good
Oh,
one. They're all going to be good ones. I'm looking forward to every one of them.
yeah, assuming everyone will show up. No. Yeah, as I said, I'm always nervous. I don't know why still, but yeah, I got confirmation from most of them, and so yeah, I usually send out emails like a couple of days before again to remind everyone. But I met with everyone, so I guess yeah, it will be happening, and will be another exciting week.
Absolutely. Be there
Great.
or be square.
Yeah, it's hard for me to make the 9 to 5 on the East Coast time slots, but some of the I have to catch replays on some of them. But that's why I enjoy the recap, because I know I missed some. But yeah, it does sound like an exciting week.
Okay, enjoy the rest of your Sunday or wherever you are. Maybe it's already Monday. So, and yeah, hear you all back soon for another cool week with Science Society. Bye, everyone. Thank
Thanks,
you.
everyone. Bye.
Thanks for joining us, everybody. Bye, Serena. Bye,
Bye.
Katerina.
Bye.
Bye.
Sorry, I just left without closing the room. I'm so sorry. Bye, everyone.
Bye.